Okay, so we're recording this thing. And this recording eventually will go live, so I have plenty of time. I'm not going to talk much about the anatomy here. I'm going to highlight a few things. So there was some confusion about the pericardial layers. Remember, there's a fibrous pericardium, the outermost, and then there's a layer of parietal and visceral pericardium. So visceral covers the heart from the outside. Between the visceral and parietal, there is a pericardial cavity. And the cavity is filled with the fluid, and inflammation of pericardium is pericarditis. Volume of the fluid in the pericardial cavity, in this case, increases, which leads to the pericardial tamponade. Okay, so that's pericardial tamponade. Pericardial friction rub is when roughened surface of the visceral pericardium starts to rub against the parietal pericardium, and surface is roughened because of the inflammation. Pericardial tamponade decreases the contractile capacity of the heart. Another term for epicardium is pericardium. Myocardium is a muscle layer of the heart. It consists of muscle. Inflammation of myocardium, myocarditis. Endocardium, layer of simple squamous epithelium that lines the heart on the inside. Inflammation of endocardium, endocarditis, and it may lead to incomplete closure of the cardiac valves and regurgitation. So blood flow through the heart. Okay, let's go over it once again. So right atrium receives the blood, deoxygenated blood from the systemic circuit. Pumps it through the tricuspid valve, from the vena cava. Pumps it through the tricuspid valve into the right ventricle. Right ventricle pumps the blood into the pulmonary trunk through the pulmonary valve. From pulmonary trunk, blood enters the pulmonary arteries. The part of it's still deoxygenated. It's the part of the pulmonary circuit. Blood reaches the lungs, getting saturated with oxygen, turns back to the left atrium, comes back to the left atrium through the pulmonary veins. Left atrium squeezes the blood, the mitral valve, into the left ventricle, and left ventricle pumps that oxygenated blood through the aortic valve into the aorta, into the systemic circulation. So basically, these questions, all of them focus on that, what are ventricles? Separated by, that all kind of stuff. No blood flow through the heart. Just know it. What lies in the coronary and interventricular sulci, different coronary arteries. Chordae tendineae, connective tissue that attaches the atrioventricular valves, mitral or tricuspid, to the papillary muscles. And the function of papillary muscles and chordae tendineae to open and close the valves. That goes like this is an extensive review of the blood flow through the heart. What separates the valves? What separates the atria and the ventricles? Atrioventricular valves. Which valve separates, here, semilunar, pulmonary, and aortic. Arteries take the blood away from the heart, veins to the heart. Veins do not always carry deoxygenated blood because, for instance, pulmonary veins actually carry oxygenated. Same is true for arteries. Valvular stenosis is narrowing. Anyone has a question? Question? Oh, Delian, I'm going to mute you. So valvular stenosis is narrowing down. Incompetent valve is when the valve cannot close completely. Treatment options. Yeah, you can replace the valve with the artificial valve or cadaver valve or pig valve. This one. Any question? I don't think you have any questions here. It's pretty straightforward. Blood volumes, same. Pulmonary, for God's sakes. Pulmonary circuit, that's oxygenation of the blood. Systemic circuit, delivering the blood to all the organs. Pulmonary circuit is shorter, has a lower blood pressure, and that means that right ventricle that pumps the blood into the pulmonary circuit has a thinner layer of the cardiac muscles. Mismatch between the volumes in these two circuits, that leads to the congestion, congestive heart disease. So we're going to go back to it when we talk about congestive heart disease, but basically then you have more blood in one of the circuits, increased pressure in one of the circuits, and associated problems. Coronary circuit, it's blood vessels of the systemic circulation that deliver blood to the heart, cardiac muscle. Angina pectoris is the condition when coronary vessels are obstructed, blood flow to the heart is insufficient, and heart becomes hypoxic, and that leads to the chest pain. Coronary arteries arise from aorta. Left coronary artery supplies left side of the heart, like left ventricle, left atrium. Right coronary artery supplies right ventricle, right atrium. Anastomoses are connections between the coronary arteries that provide additional pathways for the blood to flow and then allow blood to get around a potentially blocked small artery. They cannot circumvent the blockage of a large artery, like left coronary or something like that. Delian, you have a question? Delian, do you have a question?
No.
Okay, good. Since everybody's muted, you can write the questions in the chat. Okay. Myocardial infarction, the block of the flow of the coronary vessels, can be caused by constriction of the blood vessel, an atherosclerotic plaque obstructing the blood vessel, or a thrombus being stuck in the coronary blood vessel. Cardiac muscle involuntary contracted, striated. They striated because they have sarcomeres. They are branched, one or two nuclei. Intercalated discs contain gap junctions and desmosomes. Gap junctions provide the electrical connection between cardiomyocytes together. Okay, calcium initiates the contraction by binding to troponin. Troponin shifts tropomyosin from the actin. Tropomyosin blocks binding site on the actin, preventing the formation of the cross bridge between actin and myosin. Actin forms thin filaments. Myosin forms thick filaments and forms cross bridges. The myosin heads form cross bridges with the actin. So contractile mechanism, that's your, cardiomyocytes depolarize calcium channels in the endoplasmic reticulum, open, or calcium flows from the outside of the cell. Calcium binds to troponin. Troponin shifts tropomyosin away from the actin, so myosin can form cross bridges. And I don't know if you see me because I cannot see myself. Let me move to my. Can you see me? Okay. Sliding mechanism. So thin filament slides against the thick filament. That's the contractile mechanism. Okay, so same. If there is an electric signal, then T-tubules will transmit electric signal, opening calcium channels in the endoplasmic reticulum. Cross bridge. So cross bridge is formed between the myosin and the actin. Which molecule is required for the cross bridge cycle? ATP. So depolarization of cardiomyocytes, sodium. At max depolarization, voltage-gated sodium channels close. potassium and calcium channels open. Calcium channels are slow, but since calcium flows into the cell, they allow to maintain depolarization for a while, maintaining plateau, maintaining cardiomyocytes in the contractile state, and that makes the heart to contract for a longer time. Potassium channels repolarize cardiomyocytes. Absolute refractory period is the period when nothing can cause another contraction of cardiomyocyte, and it's important because then it prevents tetanic contractions. It happens because sodium channels doesn't just close at the maximum depolarization, they are actually getting inhibited. Metabolic requirements, aerobic respiration, okay, they need ATP. They can use lactic acid, fats, carbohydrates, actually anything. Aerobic metabolism is preferred. In ischemia, cardiomyocytes have to switch to fermentation. And that fermentation leads to the accumulation of lactic acid. Lactic acid lowers the pH in the cell. That leads to the release of calcium from the endoplasmic reticulum and mitochondrial damage. Mitochondria damage, they cannot produce ATP efficiently, so gap junctions that rely on ATP production close, and the result of that closure is fibrillation of the heart. If you have a question, I'm going to take pauses from time to time because I know that you sometimes are hesitant. If you have a question, let me know now, write it in the chat. You can select question and I will unmute you. I think we're good. Okay. Pacemaker cells, they set the cardiac rhythm. Gap junctions enable communication between smooth muscle cardiomyocytes to support cardiac rhythm. Pacemaker potential is formed. Oh, come on. Pacemaker potential is formed by calcium. Sorry, no, not calcium. Sorry, sodium. Pacemaker potential is formed by the sodium, influx of sodium through the slow leaky sodium channels, flows into the cell through slow leaky sodium channels. When pacemaker potential reaches the threshold, calcium channels open and calcium starts to flow into the cardiomyocyte. And that leads to a very rapid depolarization and action potential. Pacemaker cells are repolarized through the efflux. Potassium flows from cardiomyocytes outside. So when cardiomyocytes reach the action potential, calcium channels close. And when cell is completely repolarized, potassium channels close as well, and pacemaker potential starts again. Structures that represent conduction system of SA node, AV node, bundle of His, Purkinje fibers. The order of impulse is from SA node to AV node to bundle of His or atrioventricular bundle to Purkinje fiber. The only electrical connection between atria and ventricles is AV node. It's important because the impulse that originates in SA node is delayed at the AV node. So atria contract first and ventricles contract second. If atria and ventricles would be connected by gap junctions, then there would be no directed signal transmission, electrical signal transmission from atria to ventricles, and they would contract altogether. So the AV node, the impulse is delayed. The logical importance, as we already said, is to make sure that atria contract first and ventricles contract second. And the electrocardiogram allows us to measure the electrical activity in the heart. So when we see P wave, that's the impulse originated in SA node, leads to depolarization and eventually contraction of the atria. In QRS complex, what we see is repolarization, essentially relaxation of atria, and depolarization, essentially contraction of ventricles. And finally, in the T wave, that's the repolarization of the ventricles. So in regards to this, basically, to which extent you should know, okay, let me go here to our cardiovascular system. We can see my screen. Well, I hope, definitely hope you can. So this is the slide that I'm talking about. Basically, this 6 things that you should know. Okay. Okay. So, like, what's the PR interval? That's the time that it takes to go from depolarization, starting depolarization of atria to starting depolarization of ventricles. Why is ST segment important? Because it tells you how long your ventricles are contracted. Why is QT interval is important? It shows you how long the depolarization, repolarization of ventricles go on. Okay. Mm-hmm. So medulla regulates the heart rate. Parasympathetic sets up normal heart rate. It inhibits cardiac conduction system. Tachycardia, rapid heart rate, can be caused by influx of calcium, epinephrine, blood loss. Bradycardia, reduced heart rate, can be caused by acetylcholine or shock, massive vasodilation, calcium, Can this too be normal? Yes. Tachycardia in sports, normal. Bradycardia in athletes at rest, normal. Is the SA block? Okay. So you see these graphs. I may show you one of these graphs and ask you, this is an example of, I don't know, SA block or heart block, AV block. Describe what you see there. Describe what's there, okay? In SA block, SA node does not produce an impulse. You don't have P wave, okay? Now, if you don't have a P wave, SA node is not a pacemaker anymore. So what takes over as a pacemaker? AV node. Which rhythm is established? Junctional rhythm. Can it happen in an otherwise healthy patient? Now, junctional rhythm is not normal in a healthy patient, but can you see once in a while SA block in a normal patient? Sometimes, yes. If it's chronic and consistent, then you have a problem. Patient with a problematic heart. AV block is dysfunctional AV node. So first degree, just to slow down transmission through the AV, delay of conduction. So PR interval is increased compared to normal. In second degree, dropped beats. Sometimes you just don't have QRS and QRS complex and T wave. And in the third degree, complete block. So you have this rhythm originates in the AV node or worse. And that is really slow, and treatment option, the only treatment option here is pacemaker. So again, what you can expect, I show you the picture saying this is the AV block. What can you see on ECG? what's going to happen to conduction system, describe which changes will happen in the conduction system of the heart, blah, 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 stuff like that. Topic focus, extra beat. So that's here, you see, premature beat, premature contraction. Usually is located in the wall of the atria or wall of the ventricle outside of the conduction system. Premature contraction, this can happen in the healthy patients. Premature ventricular contraction, nicotine, ventricular or atrial, it's fine. Can it be predictor for the clinical complication? Talking about the ventricle, premature ventricular contraction, yes. Here, okay, high risk for ventricular fibrillation. Nope. Atrial fibrillation, asynchronized contractions of the atrium. The person can survive, yes. Requires pacemaker, yes. And ventricular fibrillations is a complete chaos in the ventricular muscle. Basically, heart doesn't pump the blood anymore. That requires cardiac resuscitation. Clinical complications associated with the atrial fibrillation, increased risk for thrombosis. Happens to a patient with a ventricular fibrillation, blood doesn't flow, and the treatment option is to shock them with a pants. Okay. Before we move any further, any questions? Write it in the chat if you have. No, we're good. Okay, awesome. So next series of questions are about the cardiac cycle. So let's spend some time on it. So you have a diastole. The whole heart is completely relaxed. Nothing is depolarized. Nothing is contracting. Ventricles are filling the blood. Ventricles are filling with the blood. Blood freely flows into the ventricles. That's ventricular filling. AV valves are open. SL valves closed. Now, during atrial systole, atrial contraction, atria push the blood into the ventricles, the rest of the blood. And then ventricles start to contract, and that's the moment when that is during atrial contraction, that's atrial systole, and that's when you see, right before that, you see the P wave. P wave initiates that contraction. Then you see QRS complex, atria start to relax, but ventricles contract, and that contraction increases the pressure in the ventricle and closes the valves. Okay, the semilunar valves were closed anyway, but now AV valves close, and that's when you hear the first cardiac sound. So this is isovolumetric contraction. The volume of the blood doesn't change. And then when the pressure in the ventricle becomes higher than the pressure in the vessel, blood goes, the semilunar valves open, blood goes into the great vessels. That's the ventricular ejection phase. Eventually, the blood pressure in the great vessels becomes higher than in the ventricles. So semilunar valves closed. Remember, those valves, they still closed. Okay. It becomes here, the pressure here or in the aorta is higher than the corresponding ventricles. Semilunar valves closed. All valves are closed. That's isovolumetric relaxation. And then ventricles completely relax. Heart is in a full diastole, and ventricular filling begins once again. Okay. That's your cardiac cycle. And It basically goes over each step in a very, very detailed manner. So you understand the pressure relationships, the valves that are open and closed. I honestly don't remember what I had in the practice questions for this. I think I had something. So yeah, there's definitely, you can expect, it's such a huge concept. There's going to be one of the questions on the exam is going to be about cardiac cycle. So volume of blood in the ventricle at the end of diastole and diastolic. Volume of blood in the ventricle at the end of systole and systolic. That's basically what we just covered. Cardiac output, amount of blood that heart pumps in a minute. Okay, disregard this stuff. Just disregard it. It's like disregard. So what happens to it if heart rate increases or decreases? If heart rate goes up, cardiac output goes up. If it goes down, cardiac output goes down. Stroke volume goes up, cardiac output goes up. Stroke volume goes down, cardiac goes down. Stroke volume is the amount of blood that heart pumps in a single contraction, single cycle. When EDV increases, stroke volume increases, cardiac output increases. If ESV increases, stroke volume decreases, cardiac output decreases. Cardiac reserve, the difference between the amount of blood that heart can pump. Could you draw the picture to answer the question about the cycle? I would love you to be able to draw the picture, but I'm not sure that Blackboard allows it. So you will have to write. You'll have to type. But look, how much cardiac cycle takes here? A page of questions. If I would ask you to describe cardiac cycle, dude, that would be insane. Okay. Also, if you would think, oh, he's definitely going to ask us the whole cardiac cycle. If I would be you, you know what I would do? In this case, if I would do that, I would write down the description of the cardiac cycle and then memorize it. And that's the last thing I want you to do now. So I will ask you about the cardiac cycle, but I'm not going to tell you what I will ask you about the cardiac cycle. I promise that the answer can easily fit in 4 to 5 sentences. Did I, okay, Lynn, I'm going to unmute you. Did I answer your question?
Yeah.
Okay, I don't know how it makes you feel, but
Figure it out.
I know that you cannot draw. Drawings help, but believe me, 4 to 5 sentences. I mean, if you're really wording. And yeah, it's not going to be enormous. Again, my goal is not to drown you.
Is the test gonna,
Say again?
is the test going to be?
Yes. You're going to have 90 minutes, an hour and a half.
Okay.
That is plenty of time. No, seriously, plenty of time.
Okay.
Okay. Okay, we got this. This one. Okay. Cardiac reserve, the difference between the amount of blood that heart can pump during athletic activity and the cardiac output at rest. What can increase EDV? EDV can be increased by increased preload and increased contractility. You need to know. What's going to happen to cardiac output and stroke volume if EDV or ESV increased or decreased? That's what we just answered here. I see that some of you have poor connection. So for now on, let's do this. I suggest if your connection is relatively poor, turn off the video and let me know if you have a question either by turning on your microphone or typing in the chat, okay? So blood pressure, what effect does it have on end systolic volume? What effect does contractility have on end systolic volume? So if you have elevated pressure, somebody has a question? Lillian?
Yeah.
Question?
Mm-mm.
Okay, I'm going to mute you then. So blood pressure, if it's elevated, then ventricles have hard time pumping blood out, which means that end systolic volume will go up. In case of increased contractility, it means that the heart squeezes the blood harder, squeezes out more, so less is left in the heart, so ESV will go down. Only sympathetic system can affect contractility, it can increase it. So length of diastole. If you have longer diastole, heart has more time to fill up with the blood. That increases venous return. It increases end diastolic volume, stroke volume, and cardiac output. So preload is the ability of the heart to contract harder when the cardiac muscles are stretched more. So when preload is increased, basically when the amount of blood that goes into the heart is elevated, heart stretches more, but contracts harder, therefore increasing cardiac output. Cardiac muscles, as they stretched, they can contract harder. So increased venous return and increased length of diastole, increased preload, increased cardiac output. Exercise increases preload because of the muscular pump and respiratory pump. When you exercise, your muscles contract, squeezing your veins, increasing venous return. When you breathe harder, respiratory pump expanding, chest draws the blood through your veins towards your heart, increasing venous return. Higher preload increases EDV. Contractility measures the strength of the contraction of the cardiac muscle. Brain fart, I'm sorry. Strength of contraction of cardiac muscle at the given. It can be increased by so-called ionotropic agents, agents that increase influx of calcium in the heart, okay? So positive ionotropic agents increase contractility, negative ionotropic agent decrease it. So calcium, disregard second calcium. So these two 4 agents here, calcium, thyroxine, epinephrine digitalis, they are positive ionotropic agents. And here's the cool part. I can write down it here. Okay. And obviously, these ones are negative ionotropic. Okay. Higher contractility reduces ESV. Afterload basically reflects the blood pressure in the great vessels in pulmonary trunk or aorta or both. Higher afterload means higher blood pressure. Higher afterload affects ESV by increasing it. So since blood pressure, since heart works against the higher pressure, more blood is left in the heart, ESV increases, stroke volume decreases. Before we move on, any questions?
Can you say that last part one more time?
About afterload.
Yeah.
Okay, can you see me?
Yeah.
I'm gonna do something. I can't see anymore. I'm gonna stand up for a second. Can you still see me?
Yeah.
Okay. Stay on the mic because I need a feedback. So you have a blood vessel. Okay, you have a blood vessel. Blood vessel. There's a lot of pressure in that blood vessel. When heart goes into that blood vessel, when blood goes there from the heart, it has it experiences high resistance of the flow. Does that make sense?
What? If there's
So when heart pumps blood out, blood goes into, let's say, aorta. Are we clear? Let's talk about, okay. If pressure in aorta is very high, left ventricle has to work against that high pressure. Is that clear?
yeah.
Which prevents a lot of blood from being pushed in the aorta.
Okay.
Is that clear?
Yeah.
So that blood stays in the left ventricle in the form of ESV instead of being pushed in the aorta, which subsequently increases end systolic volume. Is that clear?
Yeah.
Anybody else? A question here? You can unmute yourself and ask it by the voice or, okay, good. Okay, sympathetic stimulation increases heart rate, parasympathetic stimulation decreases the heart rate. Agents that increase heart rate are positive chronotropic. Agents that decrease heart rate are negative chronotropic. Trying to figure out how to work while standing. Calcium, epinephrine, norepinephrine. So like these ones, positive chronotropic. These two, negative chronotropic. Okay, back to sitting. Congestive heart failure, mismatch in the pumping capacity between two sides of the heart. Risk factors, atherosclerosis, high blood pressure, obesity, So basically one side becomes weak, doesn't pump blood as good as another side. So if left side fails, then congestion happens on the right side. Right side is the pulmonary circuit. So blood gets congested around lungs, and the main symptoms is pulmonary edema and pulmonary hypertension. When left side fails, congestion happens in the systemic circuit. So peripheral edema and the hepatic portal hypertension. Atria are connected by the foramen ovale. It's not a pathology in the fetal heart because you don't need to separate the circuits in the fetal heart because fetus receives oxygen through the placenta. Now, septal defects. What you need to understand, you may have an atrial septal defect and ventricular septal defect. That's it. Transposition of the great arteries is when pulmonary trunk is attached to the left ventricle and aorta is attached to the right ventricle. So therefore, two circuits are completely isolated and it's incompatible with neonatal life because there is no proper, so blood from pulmonary circuit never reaches the systemic circuit and vice versa, so there's no oxygen going into the systemic circuit. Coarctation of aorta and aortic stenosis are basically two forms of the same condition when aorta is narrowed down. Stenosis usually occurs at the longer patch of aorta. It's overall like this, while coarctations is usually a very narrow fragment of aorta that's like this instead of being this. General symptoms. So usually in coarctation of aorta, aortic stenosis, blood pressure in the upper limbs and the head is increased because blood gets congested there, while the blood pressure post stenosis, so lower limbs and abdomen, is usually lower. Cyanosis, of course, happens because not enough blood gets to, say, lower limbs. Myocardium enlarges as we age. Aortic walls become thicker. Atheromas are formed, so atherosclerotic plaques. Aorta dilates a little bit, calcifications. So there are calcifications in aorta, which makes it less elastic. And left ventricle with age, since muscles become thicker, that may lead to mild cardiomyopathy, so dilation of the left ventricle. Problems associated with the valves, they don't close completely. Okay, before we move on to the blood vessels, does anyone have questions?
So when you said that when the left side of the heart fails, it affects the pulmonary circuit.
Yes.
Why?
Think of it this way. So I have two sides. Trying to think if I can draw it or not. Okay, I think I can. Give me 1 second, okay?
Or
Let me just end the show. There we go. Where's my magic pen? Okay. You still can see my screen, right?
yeah.
Cool. So right side, left side. Okay. So that's going to be volume 1, volume 2. Normally, when everything's fine, V1 and V2 volumes that are pumped through the right side, which is pulmonary circuit, and left side, which is systemic circuit. These volumes that are pumped through the pulmonary and systemic circuit, how do they relate to each other?
They're the same.
If your question was about the left side failure, right?
Yeah.
So if left side fails, what happens to V2?
Goes down.
But blood doesn't leak anywhere, so it goes down. What happens to V1?
It goes
Okay.
up.
Yes, it goes up. And how do they relate to each other now?
The right side is greater.
Does that make sense to you?
Yeah.
Okay, yeah. So since now you have blood in the pulmonary circuit, the pressure in the pulmonary circuit goes up. That's pulmonary hypertension. And since blood vessels, the capillaries in the lungs, the pulmonary capillaries, are relatively thin, they start to leak and you have pulmonary edema. Did I answer your question?
Yep.
Awesome.
Thanks.
Sure, yeah. Okay. Arteries away from the heart, veins, heart, order of tunics, vessel wall from the inside. Anyone has a question? No, we're all good? If you have a question, just unmute you and ask it. Veins to the heart, order of tunics, from the innermost is intima, then you have media, then you have externa. Main component of tunica intima is endothelial cells, basically simple squamous epithelium, the most important. Tunica media is smooth muscle, and tunica externa is connective tissue. Capillaries do not have tunics, they just have endothelial cells in the basement membrane. Three types of arteries: elastic are just muscular in the middle, arterial the smallest. Question? Okay. Lana, you asked me a question over the email. There is an answer in the chat. It's going to be on Wednesday. Okay. Exam is going to be on Wednesday.
Okay, thank you.
Sure. Contractile, muscular arteries and arterioles. Elastic absorb pressure changes. So muscular arteries and especially arterioles have active vasomotor responses, meaning that muscular arteries and arterioles can actually dilate and constrict. Three types of veins: postcapillary, venous veins, and sinusoids. Postcapillary veins are the smallest. Veins do have vasomotor responses, but usually it's kind of larger ones. They are capacitance vessels. That means that about 60% of all the blood that we have in the circulation at any given moment is in the veins. Sinuses are the special type of veins found in the cerebral circulation. They flattened, kind of, you know. Veins do not have a lot of smooth muscles and function of the venous valves to prevent the backflow. Okay. Three types of the capillaries. Sorry, continuous capillaries, fenestrated, and sinusoid. Most penetrable are sinusoid, least penetrable continuous capillaries. Each capillary type. So this is the closed list. And do not worry about adrenal medulla, for instance. So if you know that skin, muscles, brain, continuous, small intestine, kidneys, fenestrated, liver, bone marrow, spleen, sinusoid, you're good. Okay. Which organs have no capillaries? Ligaments, tendons, structures in the eye. Functions: exchange of gases and nutrients. Main ways of transport across the capillary wall: fenestrations, pinocytosis, diffusion, of course. Capillary diameter is determined by the diameter of the blood cells, so they can squeeze through. Tight junctions in capillary endothelium make sure that nothing gets between the endothelial cells from the capillary into the peripheral tissue. Capillary bed is the set of the capillaries that supply a particular part of the tissue. Terminal arteriole supplies the capillary bed, and postcapillary venule drains it. Vascular shunt. Got questions about it. So this whole thing is the vascular shunt. This one. Vascular shunt consists of the mid-arterial and thoroughfare channel. Mid-arterial refers to arterial end here. Thoroughfare channel refers to the venular end. Okay. It is an anastomose. So anastomose, like this, these are anastomosis, short blood vessels that connect, say, an arterial and a venule, or two arterioles or two venules. Function of the precapillary sphincter is to regulate the blood flow through the capillary. If they open, blood gets into the capillary. They closed, blood doesn't get into the capillary. What can regulate the blood flow through the capillary bed? Tissue requirements. Okay. Concentration of oxygen, concentration of CO2, pH, myogenic responses, which we'll get to. Hormones. Systemic blood flow. Systemic blood flow is the blood flow through the entire systemic circuit. Volume of blood that goes through the entire systemic circuit in 1 minute. So essentially, systemic blood flow here is equivalent to cardiac output. Flow question? Okay. Blood flow through the organ is amount of blood that goes through an organ in a minute. Okay. So systemic blood flow is equivalent to the cardiac output. We've got it. Blood pressure is the pressure. We measure it in the systemic circuit usually. It's of measurement, millimeters of mercury. So the higher blood volume corresponds to the higher blood pressure. Now, blood pressure gradient, I really want you to understand this. Blood pressure is the highest in the aorta, basically at the heart. Like when heart pumps, that's the highest blood pressure. It's the lowest in the right atrium, basically. Okay, or at the entrance to the right atrium. It's basically zero. So you have to understand that what we see for blood pressure. So this is the aorta, and this is right atrium. Okay, so this is why blood goes from aorta to the right atrium. This is blood pressure gradient. Peripheral resistance is resistance to flow. How do they relate to each other? So blood pressure is directly proportional to blood flow and to the resistance. So these equations here, okay, you probably can see them. Four main factors that influence peripheral resistance. These are the ones: viscosity, length of the vessel, turbulence of the flow, vessel diameter. Remember, only one factor can be changed really quickly. It's the diameter of the vessel. Okay. So the smaller the vessel, I'm talking about this, the smaller the vessel, the higher the resistance. Blood pressure is the highest in the aorta, lowest in the right atrium. The steepest drop of blood pressure is in arterioles. Systolic pressure is pressure at the systole when blood contracts. Diastolic pressure is when blood relaxes. Pulse pressure is the difference. Mean arterial pressure, diastolic pressure plus 1/3 of the pulse pressure. If blood pressure increases, capillaries may start leaking. Mechanisms that promote venous return: muscular pump, respiratory pump. We already described them. Muscles squeeze veins, pumping the blood out. You inhale, expand your chest. blood goes up. Function of venous valves to prevent the backflow. Venular constriction does exist. It does not play a major role in the venous return. Increased blood pressure in the exercise. You have sympathetic stimulation, which leads to vasoconstriction, also increased cardiac output, increases the blood flow, therefore blood pressure goes up. Okay, so muscular respiratory pump essentially increase cardiac output, increasing blood flow. Sympathetic activation in blood pressure changes in exercise. Vasoconstriction, increased contractility, so it increases cardiac output. Elements of the reflex arch triggered by decrease in blood pressure. When blood pressure, when blood pressure goes down, reduced pressure, reduced stretch of the arteries is sensed by the mechanoreceptors in aorta and carotid arteries. These mechanoreceptors send the signals through the sensory nerves into the medulla oblongata, and they activate cardioacceleratory and vasomotor centers. Cardioacceleratory center increases the heart rate and contractility, that's sympathetic part. And vasomotor center, also sympathetic, constricts the blood vessels. Okay? So increased sympathetic activity, increases contractility, increases heart rate. Parasympathetic stimulation is inhibited. Sympathetic system controls vasomotor center. How can a hypothalamus regulate blood pressure during stress? It stimulates medulla oblongata to further stimulate heart rates and contractility. Elevated CO2, decreased oxygen, and decreased pH affect cardiac and vasomotor activity. All of these things stimulate sympathetic activation. Okay, so chemoreceptors here, elevated CO2, decreased pH, decreased oxygen, stimulate cardioacceleratory center, which increases the blood flow and increases the blood pressure. Epinephrine, norepinephrine, they stimulate cardioacceleratory vasomotor centers, increase blood pressure. Angiotensin 2 ADH effects. Both of them, angiotensin 2 and antidiuretic hormone, increase blood pressure through two mechanisms. Angiotensin 2 can stimulate aldosterone release, which will increase the retention of sodium, which will increase the retention of water, increase blood volume and blood pressure. Also, angiotensin 2 leads to the vasoconstriction. ADH can increase the water retention, increasing blood volume, and can also increase vasoconstriction. So those are two underlying mechanisms. Renal mechanisms. Direct mechanism, your blood pressure increases, blood flow through the kidneys increase, production of urine increases, you pee out more fluid, your blood volume goes down, your blood pressure in response goes down. So that's direct renal response. That's step by step. Renal angiotensin aldosterone system. So low blood pressure activates renin. Activated renin activates angiotensin 1. Angiotensin 1 is converted into angiotensin 2. Angiotensin 2 stimulates release of aldosterone from adrenal cortex. Aldosterone increases sodium retention. Increased sodium retention increases water retention. Increased water retention increases blood volume. Increased blood volume increases blood pressure. I think we have this. Yeah, here. Okay. So it shows you here renin, angiotensin 2. Angiotensin 1, angiotensin 2. And angiotensin leads to the increased blood volume and increased blood pressure. Aldosterone. And angiotensin leads to vasoconstriction. ACE inhibitors inhibit the of angiotensin 1 to angiotensin 2. Therefore, if no angiotensin 2 is produced, no aldosterone is secreted, so that reduces the blood pressure. So this one. Increased muscular pump, increased EDV, increased blood pressure, increased respiratory pump, increased EDV, increased blood pressure. Atrial natriuretic peptide. It is stimulated by elevated blood pressure. So it acts on kidneys. Kidneys start to excrete sodium with urine, which increases the volume of the urine. So you pee out more fluid, which decreases blood volume and blood pressure. Activation of renin leads to increased sodium retention, water retention, and vasoconstriction, increases blood pressure. Hemorrhage, you lose blood volume, you lose blood pressure. Dehydration, in the short-term perspective, you lose blood volume, you lose blood pressure. In the long-term perspective, your blood becomes thicker, it increases the viscosity, and may actually increase the blood pressure. Exercise, sympathetic stimulation, increased blood pressure. Angiotensin 2, aldosterone stimulation, so it's a renal mechanism, and constriction of the blood vessels, increased blood pressure. Increased body size, more vessels, longer vessels, increased blood pressure. ADH, retention of the water and vasoconstriction, increased blood pressure. Epinephrine, norepinephrine, stimulation of increased heart rate and contractility, increased blood pressure. It's all recorded, don't forget about it. But before we move on, any questions about any particular mechanism? Okay. How do we measure pulse? I mean, we squeeze the artery between the finger and the bone, basically, and we feel how it pulsates. Blood pressure, we put a cuff. By the way, why do we measure blood pressure on the arm? Any ideas? Anyone's willing to share your knowledge?
Closest to the heart.
Yeah, where would be the better place to?
Your neck, but that would like kill you.
Yeah. Ideally in the aorta, but that would be irreversible. That's how well we measured in mice. We measured blood pressure in the carotid artery. We actually introduced a little probe in the carotid artery, but that was the end of it. When we pulled it out, they dead. So when we measure it in brachial,
Question.
yeah.
About the mice. So did you ever like compare when you did it from like their neck versus like somewhere else and like what the difference was? Like how much more accurate it was?
I believe that carotid is more accurate. Another way of non-invasively measure blood pressure in mice is to measure it on their tail. They have a tail artery, but it's not super reliable.
Yeah, but like what's like how much more accurate is it from doing it like
I can't tell you the numbers. I don't know. I really don't know. Because all our mice, they were dying at the end of it was a terminal surgery. We sacrificed them. So I know that people do tail pressure, tail cuff, but I'm not aware of the comparative data. I know that carotid is very, very exact, very, very precise. Okay. Did I answer? I answered to the best of my ability, honestly.
yeah, like 7 out of 10.
Okay, sorry about that. We'll do better next time. So when we put the cuff, we squeeze it, then we basically block the blood flow. Oh, I have a question. Sometimes we have to take them on the leg. How accurate do you think that is? That is pretty accurate because you measure on the large femoral artery. Okay. So basically, if you would compare it arm, because you just want pressure is pretty much preserved when the blood goes from the aorta to the iliac artery to the femoral artery. So it's really good. If you want to measure it, say compare femoral region versus tarsal, femoral is better. If you compare brachial versus carpal, brachial is better. Okay. Jess, did I answer your question? Well, I hope I did. You can, please don't, you can, okay, thank you. Okay. Okay. So when we start to release the pressure, cortical sound appears, that's the systolic pressure. When they completely disappear, that's the diastolic pressure. Because when it disappears, there's no resistance to pumping. Clinical hypertension, 140 over 100, but I'm not going to ask you those numbers. No. Main risk factors: eating a lot of salt, having diabetes, being obese, having atherosclerosis. Okay. Primary hypertension is when hypertension is really a main problem. Secondary hypertension, let's say, are some kidney problems that in turn lead to hypertension. Hypertension, low blood pressure, again, no numbers. Acute hypertension, usually massive vasodilation or blood loss. Hypovolemic shock, you lose blood volume. Okay, you just lose blood, hemorrhage. Okay, vascular shock, massive vasodilation that leads to the substantial drop in the blood pressure, and that positive feedback loop. So drop in the blood pressure, not enough blood goes to the heart. Heart cannot pump normally. What I was talking about. Yeah, it cannot pump normally. Even less blood goes to the heart. It cannot pump normally, even less, and eventually people dead. Cardiogenic, heart insufficiency. The heart cannot pump. Heart rate drops dramatically. Or generally, cardiac output drops dramatically. Acute bleeding, tissue perfusion decreases, delivery of oxygen and nutrients to the cells decreases, carbon dioxide is not removed from the tissues, so it starts to accumulate in the tissues. Cellular metabolism goes from aerobic to fermentation, so cellular pH drops. So after bleeding, that elevated level of carbon dioxide and acidification activates chemoreceptors, baroreceptors. Baroreceptors refer to the receptors that sense the pressure. So decreased blood pressure are activated by the receptors low blood pressure. That stimulates cardioaccelerated vasomotor centers and leads to sympathetic stimulation. A question? No? You're good? Again, if you need to ask a question, just unmute yourself. What happens to heart rate and vessel? So heart rate goes up, blood vessels constrict. This is why people after the blood loss are pale. Kidneys release renin, and renin leads to the activation of angiotensin and release of aldosterone. So people start to retain sodium and water and start to pee much less. So observable outcomes of respiratory. People have elevated breathing rate to increase the oxygen supply. Heart rate goes up. People develop tachycardia. Kidneys not shut down, but kidneys significantly reduce the amount of urine that they produce. Hormones that are produced and released in response to blood pressure, well, epinephrine, norepinephrine, sympathetic stimulation. Renin, angiotensin, aldosterone system is all elevated. Sorry, these are the ones that work on kidneys. And antidiuretic hormone is produced, and it increases water retention and stimulates thirst. So people who lost a lot of blood are thirsty. So those are observable outcomes of all this activation. So people breathe faster, have a higher heart rate, they pale, and they pee very little. And responses listed above help to restore blood pressure. Cardi increases cardiac output, this shuts down the blood flow to unimportant structures like skin, and peeing less reduces the blood loss and increases blood volume. What regulates blood flow to the particular organ, tissue, muscular arteries, and arterioles? Capillaries have the slowest flow rate. The importance of it is that there's enough time for the exchange of the nutrients and oxygen. Muscular arteries change the blood flow to the organ by altering the diameter. So systemic blood pressure regulation versus local blood flow regulation. I had a couple of questions about this. So basically, here's the idea. If blood pressure is in relatively normal range, let's say if we talk about mean blood pressure, so it's like 60 to maybe 160. If it's in the normal blood range, blood pressure range, local blood will be the priority. So if blood pressure goes up, the local blood flow will adjust, maintain the normal regular flow to the tissue. But systemic blood pressure, in case of, I don't know, it being too high or too low, becomes a priority over local blood flow. So let's say here, okay? When people lose blood, the blood pressure tanks. Your organism stops caring about your skin because skin is not essential, so it shuts down the blood flow to your skin. I hope I am making sense here. So elevated CO2, decreased oxygen, decreased pH, they dilate arterioles and increase tissue perfusion. So when tissue is metabolically active, it produces a lot of CO2, which means elevated CO2 levels, consumes oxygen, which means low oxygen levels, and CO2 production and lactic acid accumulation. All of it leads to acidification of tissue and reduces pH. So in order to restore the normal chemical composition, in order to take away CO2, deliver oxygen, normalize pH, vessels dilate, blood flow to the tissue increases. Same is true for potassium and prostaglandins and nitric oxide. It all leads to vasodilation and tissue perfusion. Endothelin leads to vasoconstriction and decreased tissue perfusion. So obviously, if I ask you, what's going to happen to the vessel diameter if pH goes up? You tell me, well, and they constrict. Myogenic controls. When blood pressure goes up, it stretches out the blood vessels. And in order to maintain tissue perfusion at the same level, blood vessels shrink. On the other hand, if blood pressure goes down, stretch of the blood vessel decreases, and in order to maintain the same blood flow through the tissue, blood vessels dilate. I hope I'm making sense here because I don't have any output from you. Okay, when it's stretched, when stretch that increases in response to the not collapse, but constricts. Angiogenesis increases the density of the blood vessels and increases the blood flow to the tissue in the long-term perspective. So muscle metabolic activities. When you exercise, your muscles produce a lot of CO2 and lactic acid and consume oxygen. So CO2 increases, pH drops, oxygen drops, blood vessels in the muscle dilate and increase blood flow to the muscle tissue. Blood flow in the brain, autoregulation. When blood pressure increases, blood vessels in the brain initially will stretch. If they would stay stretched, that would increase the blood flow through the brain, which is bad because it may hurt the blood vessels. So myogenic response is that they constrict. So that process, that autoregulation, blood pressure goes up, blood vessels are stretched in response, in myogenic response constricts them, maintaining the permanent, normal, standard, constant brain perfusion. This autoregulation happens in the normal blood pressure level. If blood pressure is too low or too high, too low, they cannot dilate anymore and they just collapse. If it's too high, myogenic responses cannot constrict them. So that stretch just overcomes everything and you have massive stretch of the blood vessels, massively increased blood flow to the brain and edema. So steep drop in the blood pressure, insufficient blood flow to the brain, and you pass out. Okay. If the blood pressure in the cerebral circulation is extremely high, brain edema. Sympathetic system controls blood flow to the skin. Primary function is to regulate the temperature. Blood flow to the lungs. In the lungs, it's the opposite. So when lungs are saturated with oxygen, blood flow to the lungs will increase to pick up all that oxygen. When lungs are deprived of oxygen, blood flow to the lungs decrease. So basically for the lungs, the CO2, O2 pH changes are the opposite. For the lungs, only for the pulmonary circulation. When CO2 is down, oxygen is up, pH is up, then the blood flow to the lungs increases. Coronary vessels, when they're systolic, coronary vessels are smooshed and muscle takes oxygen from myoglobin. During diastole, blood from the aorta runs into the coronary vessels. And during exercise, your heart pumps really hard, produces a lot of CO2 and lactic acid and uses oxygen. So basically, that same metabolic regulation dilates coronary vessels. Fluid exchange. Will I ask you to calculate anything? No. Will I ask you to tell me what's going on if you have increased this, increased that? Yes. So on the arterial end of the capillary bed, fluid in general goes from the capillaries tissue, on the venular end, from the tissue to the capillaries. Capillary hydrostatic pressure is basically blood pressure. On the arterial end, it's 35, and the venular end, it's 17. Capillary hydrostatic pressure by itself moves fluid out of the capillary. Interstitial fluid hydrostatic pressure is the pressure of the interstitial fluid. It is assumed to be zero because we have lymphatic system where all excess is constantly drained to. So that's lymphatic system is responsible for nullifying. If it would not be zero, where would it move fluid? Into the capillary. It would move fluid into the capillary. Capillary osmotic pressure is exerted by the proteins, albumin, in the blood, in the plasma. It moves fluid into the capillary. Interstitial fluid osmotic pressure exerted by the proteins in the interstitial fluid. It moves fluid out of the capillary. So net filtration pressure on the arterial end is 10, and it calculations are on the slide, okay? And fluid on the arterial end goes into the tissue. Filtration pressure on the venular end is 8, and it drives, no, on the slide it's -8. Negative means that the fluid goes back into the blood. And remember, there's a difference, like 10, -8. So some of the fluid stays in the tissue and drains into the lymphatic system. So you have a constant flow of fluid through the tissues. That removes all the crap and dead cells and stuff. So what happens on the arterial and venular end if systemic blood pressure increases? NFP on the arterial end will go up, like from 10 to 11. And on the venular end, it will kind of go down, like -7 or -6. Well, again, up or down, I don't know. Basically, the difference between them will increase, and that means that more fluid will go into the tissues, and that means edema. When systemic blood pressure decreases, NFP on the arterial end goes down, say from 10 to 9, and on the venular end, it goes down, let's say from -8 to -9. So there's no difference, there's no fluid going into the tissues. Protein concentration in plasma. When protein concentration in plasma increases, capillary osmotic pressure increases, and that drives more fluid back into the capillaries, essentially decreasing NFP on both arterial and venular ends. So say from 10 to 9 on the arterial and from -8 to -9 on the venular. And if lymphatic vessels are obstructed, that affects interstitial fluid hydrostatic pressure. It increases it, and basically it moves more fluid from the tissue back into the capillary. Okay, so that's not relevant anymore. So this one, I strongly encourage you to be able to label it. This one, that's for review, you know, calcium binds to troponin, switching tropomyosin forms crossbridge. Cardiomyocyte action potential. You're not going to be drawing anything, but it's still helpful to know what is going on, okay? Why do I have 2 cardiomyocyte action potentials? Oh, you can use this grid. Sorry, that was a kind of a mistake. You can use that grid to draw the pacemaker potential. This one for studying for EKG, labeling if any waves are missing. That's your cardiac cycle. And this one is a pretty good review what happens to EDV and ESV to fill out and check yourself. Okay, we went over this stuff already pretty well. Now, before we conclude this, I want to ask everyone, do you have any specific questions that you want to ask me now? No? Okay. Jess, let's just talk. Let me just unmute you, okay? Just talk. Okay. What do you mean by making up a test?
There's a test that I missed. I never took it.
Oh. Yeah, you will make it up. If you missed a test, you will make it up. But I'm terribly sorry, but I really have to figure out how to deal with all this transition shit.
Right.
Okay, so we'll figure this out first. We'll get more or less in the groove, and then we'll think about makeups. Okay.
Okay.
I promise that's going to be fine.
Okay.
Okay.